Chapter 11, Part 2, we're going to be looking at these two you must knows, and also this you must know here. So this one, and this one. But I will say a lot of this is going to come from doing practice problems, and same with this one. So as you watch this video, I'd highly encourage you just to kind of let the information just be put in your brain, absorb the information, but don't worry if every little detail isn't completely clear or if you don't feel completely confident about doing genetics practice problems after this video, because we'll spend a lot of time in class going over it. So I really need you to understand the biology of genetics practice problems so that if you were to read a problem, you understand the vocabulary and you understand like the concepts that are happening, what's happening inside of cells and inside of gametes. But again, just kind of let the information fall on your brain and don't be overwhelmed. So based on Mendel's research with pea plants, he came up with 2 laws of heredity. Now, these laws are really important. It's important that you write them down and pay close attention as I explain them, because these laws we're going to come back to as we continue to study genetics. So first law of heredity is called the law of segregation. I'm going to read it to you and then I'm going to explain it. Here's how it goes. When gametes are produced during meiosis, homologous chromosomes separate from each other. Okay, this is a law that happens every time gametes are made during meiosis. Homologous chromosomes separate. Okay, now what does this mean? It means that if homologous chromosomes are separating into different gametes, then each allele for a trait is packaged into a separate gamete. Okay, now I just explained the definition. Let's talk about what it means. So let's say I take a diploid cell, a somatic body cell from one pea plant. Okay, so I'm taking any cell that's not a sperm or egg, any cell that's not a gamete. And I look at the homologous chromosomes for a certain gene or a certain trait. And in this case, let's say I'm looking at the color of the flower, right? On the homologous chromosome pair, right, we're just using letters to represent this, but what these means, there's 2 alleles, right? 2 pieces of DNA, one from their mom and one from their dad, with instructions for what the gene is, instructions for what the color of the plant is. And notice here we have homozygous dominant, so we have the same alleles. Now, when this plant is making gametes, when it's making its sperm and its egg cells, it's going to separate its alleles into separate cells. So remember, we talk about meiosis. So we have 2 homologous chromosomes, capital P and capital P, right? Right before meiosis, they replicate their DNA. So now we have capital P, capital P, capital P, capital P. Here's our homologous chromosomes, but each one has sister chromatids, right? So meiosis 1, we separate the homologous chromosomes, right? So we still have 2 capitals and 2 capitals. Meiosis 2, we separate the sister chromatids. So now each of the 4 gametes in this round of meiosis all have capital P alleles on this chromosome that has the instructions for color of the pea plant, right? So the only gametes that a homozygous dominant cell could create would be capital P alleles. Same thing for recessive, homozygous recessive. All gametes will be the same because this is the only allele this parent has for this gene, right? It only has little p alleles. However, if you're heterozygous, think about it this way. Capital and lowercase, let's make a copy. Meiosis 1, meiosis 2. Now you have capital P, lowercase p. But we can simplify that. There's 2 options. So 2 different types of egg cells or sperm cells, gametes. You could either donate capital P or lowercase p alleles. Now remember, organisms are going through meiosis hundreds of times and creating hundreds and thousands of gametes of sperm and egg cells. So if you're a heterozygous flower, you could either pass down the capital P allele or the lowercase p allele to your offspring. It just depends on which one ends up being passed down. And that is determined by literally, like if you're talking about flowers and you're talking about sperm, these are pollen cells, which pollen cell lands on the neighboring flower, right? So that's the law of heredity, and we can kind of look at it up close here. So if this is a diploid cell, right, we're just showing one pair of homologous chromosomes here, but remember, most cells have multiple pairs. Like in humans, we have 23 pairs, but we're just looking at one pair here. So this is from the organism's dad and mom. The DNA, right, we have homologous chromosomes. We now have sister chromatids, like you see here, 2 sister chromatids, right? And notice here that here's this locus. This is one spot on the chromosome. It has the same genes, and each one can have different alleles. So during meiosis 1, you're going to divide the homologous chromosomes. And then during meiosis 2, you're going to separate the sister chromatids. So now you have 4 gametes, and each of them has one of the alleles. Okay, so that's why this is saying each allele is packaged into a separate gamete, because this one's packaged into this one's packaged into this one. Okay. Okay, now let's look at meiosis up close and let's ask ourselves this question: which meiotic event creates a law of segregation? So why are alleles separated from homologous pairs during meiosis? So let's look here. Remember, here's our diploid parent, here's a homologous chromosome pair. Let's look at this locus on the chromosome. We have a dominant allele and a recessive allele here, so heterozygous. Right before meiosis, each chromosome duplicates its DNA, so now you have sister chromatids, but still homologous chromosomes. What happens during meiosis 1? The homologous chromosomes line up at the metaphase plate, right? Oops, like this, and then they separate into separate cells. So now you have one here and one here, and then meiosis 2, now you have the sister chromatids are separating, and now you have gametes, one capital S, and 2 lowercase s's, right? So at which event are the alleles from the original parent separated? It happens during meiosis 1. Okay, so after meiosis 1, now the homologous chromosomes separate, and now you have separated the alleles from each of the original homologous pairs. Okay, so another vocab word I need you to know is this idea of a monohybrid cross. What is a monohybrid? A mono means one, and crossing is taking 2 plants and crossing each other. And this is a word to describe one type of experiment that Mendel did. So when Mendel was looking at one specific trait or one specific character, that's called a monohybrid cross. So if Mendel was just taking one purple flower and one white flower and crossing them and just observing the flower color, we call that a monohybrid cross. If Mendel was looking at the pea plant seeds and crossed a green and a yellow, he was just looking at the seed color. That's called a monohybrid cross. Okay, she's just focusing on how one trait is passed down. However, we need to learn about this new term called a dihybrid cross, right? Di means 2. What is a dihybrid cross? Well, this is a different experiment. So Mendel was looking at different things. In dihybrid crosses, Mendel is looking at the inheritance of 2 different characteristics or 2 different traits. We call this a dihybrid cross. So for example, Mendel took peas and he looked at the seeds of the peas. But when he had them cross with each other, he was looking at how the seed color was passed from parent to offspring, but also how the seed shape. So when you're looking at 2 different traits and how they're passed from parent to offspring, that's called a dihybrid cross. 2 different traits, how they're passed down from parent to offspring. So let's look at an example of a dihybrid cross. Okay, so remember, in a monohybrid cross, Mendel was just looking at either seed color or seed shape, just one trait at a time. But now let's look at 2 traits at a time. So in this case, let's look at our pea generation, our parental generation. Remember, these are true breeding. These are purebred, round peas, and they're also yellow. So we're looking at 2 traits, color and seed shape, pea shape. In this case, we're looking at green and wrinkled. Let's make a prediction. If these are pure breeding, well, what are their offspring going to look like? Which of these is dominant, green or yellow? For that trait. And then for the second trait, wrinkled versus round, which one is dominant? So once we cross 2 purebreds, we're going to be able to see. Here's our F1. Here's our hybrid generation. Let's start with color. What color is found in the F1 generation? 100% yellow. What shape is found? 100% round. So what does that tell us about which of the alleles is dominant and recessive? Which one is dominant? Yellow or green? Round or wrinkled? Let's take a look. Well, this tells us that yellow is dominant, right? Because in the first generation, the dominant trait is taking over, even though this pea did pass down its alleles as well, but this one is dominant, it's taking over. And it also tells us that round is also dominant. So we're going to use these alleles, capital Y to represent yellow and capital R to represent round, which means that this plant here is recessive. So green is recessive to wrinkled. Now, once Mendel did this, what did he do next? Oh, this also tells us, before I get to that, the genotype, right? So we know the phenotype is yellow and round. What is the genotype? So we can kind of look first at this first gene. So we're looking at color, and we know it's homozygous dominant, right? Because it's pure breeding. And we have homozygous dominant as well for this second trait. So remember, dihybrid crosses, we're looking at two different traits at the same time. What is the genotype of this plant here? Take a moment, think about it. Remember, it's true breeding, so it's never mixed its alleles with any other plant. And we have homozygous recessive for the color and homozygous recessive for the shape as well. Okay, so what is the genotype of this next generation? It's going to be heterozygous for color and heterozygous for shape. Okay, that's because this parent passed down this allele for color, and this parent could only pass down this allele when it's passing its sperm and egg cell are coming together. This one can only make capital Y alleles, and this one can only make little y alleles. So together, you get capital and lowercase. Let's look at shape, right? This can only pass down capital R. This one only has lowercase r in their alleles. So now our homologous chromosomes for the offspring will be heterozygous. Okay? Now, Mendel then let these hybrids here self-pollinate, so it pollinated itself. And what did he find in the F2 generation, right? Let's take a look. He found something interesting. When he did this thousands of times, he found a common pattern. He found that 9/16 were yellow and round peas, so those two dominant traits together. 3/16 were green and round, so it had the dominant shape, but the recessive color. In this case, 3/16 yellow wrinkled and 1/16 green and wrinkled, so the two recessive alleles. Now, he summed that into a ratio, or he distilled that into a ratio of 9 to 3 to 3 to 1. Why are there 4 parts of this ratio here? Well, it's because you're looking at 4 different things. You could either have round and yellow, green and round, yellow and wrinkled, or wrinkled and green. So whenever you're making these ratios, think about how many total phenotypes can you see, right? There's 4 total phenotypes here. Okay, so what's going on here? Why does our F2 generation have a 9 to 3 to 3 to 1 ratio? Why is there a mix of traits? Well, let's look back at our parent generation. Here's our pure breeding yellow and round genotype or organism here. Here is our wrinkled and green, right? When this is making gametes, it can only donate capital and capital, 2 dominant alleles, and this one it can only donate its recessive. So when we're making our F1 generation, it's going to be a dihybrid where it's going to inherit a dominant and a recessive Y allele, right? So we have heterozygous and then dominant and recessive, right? Dominant from this parent and recessive from this. And so what is its genotype? It's heterozygous for color and it's heterozygous for shape, but we have our dominants are taking over, so yellow and round. But what would happen if we took this and we were to have it go through meiosis and make its own gametes? There's 2 predictions, 2 ways that it could go. Here's prediction number 1. Well, when this cell is going through meiosis, it's possible that it could, in its sperm cells, right? Imagine this is making sperm. In one of these sperm cells, remember, it can only place one of these 2 alleles into the sperm. So let's say it just places the Y. The hypothesis of dependent assortment is this. If the dominant Y is going into this sperm cell, then the dominant allele also has to go into this one. And then the recessive allele goes into this sperm cell and the recessive into here. So the hypothesis here is that this is one option for sperm and this is one option. The same thing for eggs, right? If this is reproducing with itself or crossing with itself, then you basically get the same gametes here. Now, if you do a Punnett square here, you bring down the Y, capital Y, you bring down the capital R, you bring across the lowercase, or the capital Y here, capital R here, capital Y, capital R, right? And if you do the whole thing, you work it out. We'll do some of this in class. You'll find that you get 3 yellows to 1 green for this phenotypic ratio, right? That is what we see, 3 yellows to 1 green. Same thing for the shape. We get 3 smooth to 1 wrinkled. Now, this is not what Mendel actually found, right? Mendel in his results found these numbers here. So he did it hundreds of times. He found the ratio 9 to 3 to 3 to 1 with these different character traits, right? So let's follow the hypothesis of independent assortment, right? In fact, it's that this capital Y and this dominant R can be put together in one gamete. If we're making different gametes, so you can actually take a capital Y and a lowercase r, that could be one sperm cell, or you can kind of mix it up, right? And we'll do some practice of this in class. I know it's kind of tricky to see here, but the idea here is that these sperm cells can be a mix of the alleles for each of the traits, right? Each of the genes. And then when you fill out the Punnett square, we'll do this in class, you actually find this ratio, 9 to 3 to 3 to 1. So this is actually the hypothesis that is correct, the hypothesis of independent assortment. All right, so let's take a look at how to do one of these dihybrid crosses using a Punnett square. And it's a little complex because there's so many letters here, but if you just do the same thing every time, it'll be pretty straightforward. So the first thing you want to do is you want to figure out, I'll kind of go back here and I'll draw it in here for you. You want to figure out what are the gametes going to be? What are the sperm cells? So let's put the sperm cells up here. So let's take a look at, let's say this is going to be our sperm or our pollen generator. So this flower, this pea plant is going to make the sperm. So what optional sperm can we make? Well, let's take a look first at the color. So you could actually have a capital Y, right? So maybe, remember, our sperm cells only have one copy of each allele. And then you could have a capital R, right? But what if you had a capital Y and a lowercase r? So capital Y, lowercase r. Or what if you had a capital R and a lowercase y, right? Or what if you had a capital or lowercase, and lowercase, right? So those are the optional gametes you can make, because when you're making gametes, you only have one chromosome, one of the homologous chromosomes, one copy of each allele. Same thing goes here. You could have, two dominant, and recessive, dominant, and recessive. Okay, when it's time to cross, I'm going to start with my Y's. So I'm going to go down here and just put the Y's. So I'm going to go capital Y. Actually, I'll do the R's too. And capital R, capital Y, capital R, capital Y, capital. I'll just do like one of these so you can see. Capital Y, capital R. Okay. And then you would keep going down, the same thing. And then let's go across. So let's go across here. So this capital, so you're going to add capital here, Y, capital R here, capital, let's go across this way, capital Y, lowercase r, go across here, lowercase y, capital R, and go across here, lowercase y, little r. And then you could continue going down, right? Go down this way, down, down, and across. And then what you're going to end up finding are the potential genotypes of their offspring if you cross this with this. So it's possible you could get a yellow homozygous dominant or yellow phenotype, and also a homozygous dominant round genotype and then round phenotype, right? So this is how to do a... Okay, so let's take a look at Mendel's second law of heredity, the law of independent assortment. I'm going to read the law to you, and then I'm going to explain it to you here. Make sure you have this written down. So the law of independent assortment says that each pair of alleles segregates into gametes independently. So let me explain what this means. Notice here we have a diploid cell. This comes from a pea plant. And let's look at its genotype. So its genotype for the pea color, it's going to be dominant and recessive heterozygous. So we're going to see yellow, right? Because yellow is dominant to green. For the pea shape, the seed shape, we're going to see heterozygous. So we're going to see it's going to be round because we have one dominant round allele. Now, what happens when this diploid cell is going through meiosis to make gametes, to pass its genes on to the next generation? Well, we have to think back to meiosis. Now, notice here that in this diploid cell, we have this pair of chromosomes and this pair of chromosomes. What are we looking at? Well, this right here is a pair of homologous chromosomes, same size, same shape. They came from different parents, right? They have different alleles. This one has the yellow allele. This one has the green allele. Here's another pair of homologous chromosomes. It's a different pair. This one has the instructions for wrinkled peas or wrinkled seeds, and this one has the instructions for round seeds. And this matches what we see here, right? Capital Y, our dominant allele, lowercase y, our recessive allele, our green allele. Here is our round allele, and here is our wrinkled allele. So we're looking at two different pairs. And think about it this way. If I were to take one of your somatic body cells and look inside and look at the karyotype, look at all the chromosomes lined up, right? You would see 46 chromosomes, but each come in a pair, right? And why is that important? Well, your first pair of chromosomes has thousands of genes in it, thousands of pieces of DNA, and all of those genes are the same for those two homologous chromosome pairs, right? So this might have the instructions for hair color and eye color, chromosome 1. But then if you were to look at chromosome 5, they have the instructions for maybe skin color on that chromosome. So in the same way, we're looking at different pairs of homologous chromosomes. This one has the gene for color. This one has a gene for seed shape. Now, when this cell is dividing, right, it's going to make a copy of its DNA, meiosis 1, meiosis 2. But if we kind of summarize the whole thing, we're going to end up dividing the cell in half, and you're going to get yellow. So here is our dominant allele, and then wrinkled, our recessive allele. Why is that? Well, it's because these ones are being split off into this cell. In the same way, remember we made a copy of the DNA, so we get 4 cells in our gametes, the same thing here. When this cell divides here, we are going to get our recessive allele and then our dominant for the second chromosome. So again, here's a haploid cell because it has one copy of each gene, one copy of each allele, and one copy of each chromosome, haploid. Now, here's the other thing that could happen, right? So what's happening here is these homologous chromosomes are lining up on the metaphase plate during meiosis, but it's possible that they could be assorted differently, right? So here we have yellow and green on this side, yellow and green, same thing. But in this example, what if the chromosomes were swapped on the metaphase plate for chromosome pair 2, right? What if you have this and this? So when the chromosomes, or when the cell divides in meiosis, right, you're going to get some cells with yellow and smooth, and some cells with green and wrinkled, green and wrinkled, green and wrinkled. So these are the options that could be made, either this or this. So this is called the law of independent assortment because it's talking about how the chromosomes sort each other, right, or sort on the metaphase plate. And it's called independent assortment because the way that these chromosomes are set up, right, we have the dominant on the left and the recessive on the right, does not determine how the next chromosome pair will line up. So it's not necessary that the dominant are always on the left and the recessive are always on the right, right? You can see here's our dominant and our recessive, and then our recessive and our dominant. So all the pairs of chromosomes line up independent of each other. They're not influencing each other, and that creates diversity in the gametes that are created. So let's look at meiosis again. Let's kind of review what's going on here. So if we're looking at genes on different chromosomes, right, that's important for a law of independent assortment. We're focusing on genes on two genes, right? We're talking about dihybrids, talking about two different traits, two different genes that are found on different homologous pairs, right? So here is chromosome pair 1 and chromosome pair 2. They're homologous. Each has made a copy of its DNA. We see our sister chromatids. We got some crossing over going on. And then now let's look at meiosis. So the homologous pairs are going to line up on the metaphase plate, right? Here's pair 1, here's pair 2. They've crossed over a little bit. And now this cell will divide in half, and these two will end up on this side, and these two will end up on this side, right? And then the sister chromatids will divide. This one will go here, this one will go here, this side will go here, and they'll donate one sister chromatid here. And now you have your 4 gametes. But it's possible that this could have been swapped. So the blue could have been on this side, and the red could have been on this side. And that's also going to change the way that the gametes are created. So remember, one organism does meiosis many, many times, thousands of times. And so depending on how the chromosomes line up that round of meiosis, you're going to make different versions of the gametes. Okay, so what meiotic event creates a law of independent assortment? It's meiosis 1. It's how the chromosomes line up on the metaphase plate during meiosis 1, depending on how they're sorted or assorted here. Okay, so let's walk through both of Mendel's laws again, and let's look closely at meiosis. No, this picture is very detailed, but really this summarizes and is a really great explanation for all the kind of details you need to know about the 2 laws of inheritance. Okay, so let's look closely at our P generation. Here is our phenotype. We have round yellow, and our genotype, it is pure breeding. So we have homozygous dominant for the color and homozygous dominant for the shape, right? Now, what does this mean in terms of the chromosomes? Let's look at the homologous chromosomes for the color allele. Notice here, they're kind of all jumbled here together. In reality, chromosomes are all mixed up. But here's our homologous pair 1, so capital Y and capital Y. And here's our second homologous pair. And this is a pair of chromosomes that has the instructions for the seed shape, right? So in this case, we have different chromosome pairs, and each one has a different gene on it. One has a gene for color, and one has a gene for shape of the seed. Now, when this P generation is making gametes, it can only donate capital and lowercase y's if you follow meiosis. Same thing here. When this cell is making gametes, only little y, little r. Now, let's say these gametes fertilize each other. So this homologous chromosome will pair with the R, so capital R, little r, right? One from mom, one from dad. Now you have your new homologous chromosomes in our F1 generation, in our hybrid. That's why our genotype is capital R, little r, heterozygous, because our parents passed down, this one passed down dominant, this one passed down recessive for this first chromosome pair. And then when we look at the seed shape, we have big R and little r, right? And you see in this here, that passed down. So literally those gametes come together and the chromosomes are now mixed. And we have you see big R, little r, so we have heterozygous for the seed shape. Okay, now what happens when these cells are making gametes, when they're making meiosis? Well, let's start with the first step, which is to make a copy of the DNA. So if you see here, now you have your sister chromatids. Then you have the homologous chromosomes pairing up on the metaphase plate. So here's pair 1, here's pair 2, right? And remember, here we can see the principle or the law of independent assortment. Notice dominant, recessive, that's how they're arranged. That's how they're assorted on this version. But it's also possible that we could have recessive, dominant, recessive, right? So we can see here the dad's chromosome and the mom's. Dad's on left, mom's on right, right from up here. And then here, mom on left, dad on right. So it depends on where they assort. And this is in meiosis 1. Now the alleles are going to segregate, right? So as they're going through anaphase, we have the dominant and the recessive. They're segregating, they're separating into 2 separate cells. So meiosis 1, the homologous chromosomes separate. So here's our homologous pair 1 over here. And for chromosome 2 down here, same thing here. Then we have meiosis 2. The sister chromatids get separated. And now in this version of meiosis, you can create gametes that have, again, these are haploid. They have only one copy of each chromosome now. Here's dominant, homozygous, or this is just not homozygous, just dominant for the 2 different genes, right, on different chromosomes, chromosome 1, chromosome 2. And the same thing here. Then you have these options. You have dominant and recessive, or no, 2 recessive for each gene, for each of the genes. Here you have recessive dominant and dominant recessive here. Okay. And then if you were to have these plants mix their genes, and you did this hundreds and thousands of times, you could have this gamete fertilize this gamete, or this one fertilize this one. But if you did it enough times, statistically, you would find this ratio. And if you think about why, it's because you really have more dominant alleles in the population. So dominant. And so you're going to end up getting more of this dominant shape here. All right, so that's kind of the chromosomal basis of Mendel's laws. It's really helpful if you understand this. Like if you understand this, you're really understanding heredity really well. Meiosis, alleles, genes, and the 2 laws of inheritance. Okay, so just to kind of summarize, the law of segregation, we're looking at I don't know why I'm blurry here. We're looking at for a monohybrid cross. So for a single trait, we're looking at how, okay, for this trait here, for let's just say we're talking about the seed shape, the alleles are separated in meiosis 1. Okay, so that's what we're talking about. We're talking about law of segregation for each trait. If we're just looking at one trait here. In this case, we're actually looking at 2 traits, right? 2 different. But if we were just looking at one, you would see that they're separating. Established by meiosis 1. For law of independent assortment, we're talking about like 2 sets of chromosomes or 2 or more traits. So in this case, remember, law of independent assortment says on the metaphase plate, this and this, they can be assorted, however. And again, most cells actually don't just have 2 pairs, right? We have 23 pairs in humans. And so they can be sorted either way on the metaphase plate. So make sure you understand this, and make sure you're not just memorizing it. Like go back here, read this part in the textbook if you need, so that you really, really understand this part. Okay, one last law. It's pretty like obvious, but just to go over it really quickly, it's called the law of dominance. And basically that means if you have one dominant allele in your allele pair, you're going to express that trait, or you're going to see that trait in the phenotype. So we have no dominant alleles here, so you don't see the dominant trait in the phenotype. Whereas here, we have one dominant allele, so that's what the phenotype will look like. So basically, you understand this. You probably learned this many times, but as long as you have one dominant allele, you're going to see that phenotype. Okay, why did Mendel choose peas? Just really quick. Peas are good for research. There are many different varieties, so there's totally different traits like shape of the seeds, color of the seeds, flower color, right? There's different things you can study. He also had strict control. So each pea plant has like strict male and female structure, like I showed you in the last video. And so it's really easy for Mendel to like take pollen from one flower and basically put it on another flower. It's really easy to get plants to cross-pollinate. But pea plants can also self-fertilize. So it can take its own pollen and fertilize its own egg. So he could cross-pollinate, he could move plants from one to another. So he could really control how are the gametes from one flower, or which one are they breeding with if you're trying to breed it with another flower, right? He can control a lot of things. They're really easy. They're good for genetic research. They're simple genetically. Most of the characteristics are controlled by a single gene. So if you have chromosome pair 1 and that has the gene for P color, right? There's just one gene. So as long as you have the capital P, you're going to be yellow. There are some we're going to learn later. It's called non-Mendelian genetics. There are some genes or some character traits, like for example, eye color in humans. There's not just one gene for eye color. We actually have multiple spots, about 100 different genes in our entire genome, and all of our chromosomes, our 46 chromosomes. There's about 100 genes that have the instructions for eye color. But for pea plants, they're very simple, just one gene for the pea color. And each gene only has 2 alleles. So sometimes you might have more than 2 alleles. So instead of like yellow versus green, there's actually like yellow, green, and purple, right? And that would be harder to study. So it's easy to see like which one's dominant over the other because there's only 2 alleles for each gene. So let's review Mendel's laws. First, the law of segregation, the idea that alleles segregate, they separate when gametes are being formed, sperm and egg, and independent assortment, when homologous chromosomes are lining up to divide through meiosis. It doesn't matter if the dad or the mom's chromosome is on either side, they can line up independently. This homologous pair lines up independently from this homologous pair. And we can actually do some probability and some statistics on this. And we can combine genetics and probability. And we can use some laws of probability, kind of similar laws that you would think about when you're rolling dice or tossing coins, to kind of do some math and make predictions based on what you know about alleles and genes. So let's start with some calculations. So we are going to be calculating the probability of making a specific gamete, and it's kind of like calculating the probability of flipping a coin. Okay, so let's say you have an organism and it has a certain gene, right? So let's say we're talking about this gene, and let's call it flower color. Okay, so we're talking about flowers, we're talking about flower color, and here's one cell from a flower. If we were to zoom in, you would see homologous chromosome pairs, but we're just abbreviating with letters, one dominant allele, one recessive allele. Now, when this organism is making gametes, when it's going through meiosis, if you follow the steps of meiosis, right, meiosis 1 and meiosis 2, the only possible gametes are going to be capital P or lowercase p. These are the alleles that are found in the gametes. But what is the probability that your gamete is going to have a capital P versus a lowercase p? So let's think about it. Well, we only have 2 options, so we actually have a 50-50 chance. In the same way, let's say we have a capital P and a capital P here. What are the chances that you're going to get a capital P allele? What are the chances you're going to get a lowercase allele in the gamete? It's 100% capital, right? No chance that you're going to get a lowercase p allele here. Now, we can kind of think about this how in the same way that we would think about tossing a coin. And so here's the kind of probability law that you need to know. When you're tossing a coin, if you toss a coin and you get heads or tails, that outcome doesn't have any impact on the next toss. So if you toss a coin and you want to know what is the probability of getting a heads, and let's say you do get a heads, that result is not going to affect if you were to flip it again, right? So probability of tossing a heads each time is 50-50, right? It's random. In the same way here, we're looking at the possibility of making a p gamete each time. It's 50%, okay? So we can use this thing called the rule of multiplication that helps us kind of quantify what's happening and make predictions. So here's what the rule of multiplication says, and I encourage you to write this down, and I'll explain it. It's this idea. It's a chance that two or more independent events will occur together. So it's a probability. Like, let's say you toss a coin and you see if it lands heads or tails. And then you toss another coin and you want to see if it also lands heads or tails. So whenever you think of multiplication, you're thinking, what's the probability that this will happen? What's the chance that this will happen? And what's the chance that this will happen? Okay. Now, here's what you do. You're going to take the probability, the chance that, let's say we're talking about flipping a coin. What's the chance that you're going to get heads up the first time? It's a 50-50 chance, so 1/2. But then if you want to know the possibility that you would get a heads up again, you multiply those, and then you get 1/4. So here's how I think about it, right? I think about it in words. Whenever you hear the word and in probability, you're going to multiply in this case. Okay, so if I say these words, listen carefully. What's the chance that if you flip a heads up once, you will also flip a heads up again? What's the probability that you would flip a heads up and another heads up, right? So you have and there. So 50% probability that you're going to get heads up once, and a 50% you're going to get a heads up second time. And so your calculation would be 1/4. Now, in the same way, let's ask the question, what's the probability that if you cross a heterozygous plant for the flower color with another heterozygous, so two hybrids, what's the probability you're going to get a homozygous recessive offspring? Well, let's think about it. These parents can donate their alleles to their gametes, and this one can make capital P, this one can make lowercase p gametes, and then this one can make capital P or lowercase p. But we're going to end up with little p, little p. Now, let's start with the first parent. What's the probability that this parent is going to donate the little p allele to the offspring? The chances are 50%, right? 50% chance that they're going to give the little p. And what's the probability that the other parent 2, will donate the little p allele? It's 50%. So when we're looking at this, it's the probability that parent 1 will donate a little p, and the probability, so multiply, and the probability that the second parent will donate the little p allele equals 1/4. Okay, so we're going to be practicing this more in class. It's just an introduction. But whenever you're thinking about the rule of multiplication, think about when two events are happening and they're occurring together. Okay, the chance that two events will occur together. So one event and another event occur, but they're not dependent on each other. One is not affecting the other. They're just both happening at the same time. That's the multiplication rule. So we can kind of look at it this way. So let's say we have two alleles, right? So here is one parent and another parent, and we're heterozygous here, heterozygous. What are the chances that we are going to get sperm cells from, let's say this is the male, sperm cells that have a capital R? It's 50%, right? 1/2. What are the chances that we're going to get little r? It's 1/2, right? So 1/2, 1/2. When we're making eggs, let's say this is a female, what's the chances you're going to get a little r in the gamete, in the egg cells? 50%, 1/2. What's the chances you're going to get a little r? 50%. Now, at this point, now you can make some combinations. So if you were to ask yourselves, okay, what's the chances that you're going to get a capital R and a capital R from these two parents when they make babies? Then what you are going to do is you're going to multiply the probability of a capital R from the sperm and the capital R probability from the egg, right? That's why you multiply, because it's 1/2 times 1/2, the probability of capital R and the probability of capital R. And so you're going to get two results, right? Two independent events are occurring, what's the probability you're going to get both of them at the same time? That's the rule of multiplication. If you were asked the question, what's the probability if you had a heterozygous dad and a heterozygous mom that you would get heterozygous offspring? Well, you would say, okay, well, the probability of getting a little r from the dad, the sperm, is 1/2, and the probability of getting the big R from the mom, 1/2, multiply them, you get 1/4. Okay, so this is how we're going to be figuring out our potential offspring genotypes. Now you're going to notice that you have the same genotypes here. It's just that this little r came from the egg and this big R came from the sperm. Whereas this big R came from the egg and this little r came from the sperm. Okay, so we're going to come back to that, but keep this in mind for now. This is kind of how you look at Punnett squares and use probability to figure out the percentage of the offspring, or the chances that you will have the offspring. All right, so we use the rule of multiplication to apply it to the dihybrid cross. So remember, a dihybrid cross would be if you're looking at the inheritance of two different genes on two different homologous chromosome pairs. So I'm going to walk you through this, but we're going to continue to go over it. It's more helpful if I kind of draw some things out on the board and show you in person. And I'll also link a video, really helpful video, to Bozeman Science, which will go over this a lot more. But I'm just going to kind of give you an introduction into it, and then you can come back and look in class and at the other videos I post. Okay, so here's the question. If you have heterozygous parents, and this is just showing one parent, but let's say you have two heterozygous parents. So I'm going to kind of use my pen here. So let's say this is dad, okay, and this is mom. So mom is the same exact. So big Y, big R, little r. And this is why I want to do most of it in class. So you're going to cross these two, right? So take this one, cross with this one. The question is, what's the probability of producing offspring with this genotype? Homozygous recessive for the color, and homozygous recessive for the seed color, and homozygous recessive for the shape of the seed, right? If we're looking at pea plants. Okay, so first, let's just focus on this parent right here. Okay, let's focus on this. First thing we want to ask, these are our offspring, right? This is our result. But we want to figure out, okay, what are the gametes that this parent right here can produce? And we want to kind of focus in on each gene at a time. So let's just focus, I'm going to zoom in even more, on this gene. Okay, so we're looking at the seed color. What is the probability that you're going to be from this parent producing a little y gamete? What are the chances? Well, it's 50-50, right? Either this one or this one. So the chances you're going to get a little y is 50%, or 1/2. Okay, now let's go back and let's actually zoom in on this guy. So let's look at this gene for this parent, right? This is still one organism. What's the chances that this flower is going to produce a little R gamete? Well, it only has two options, and so it's a 50-50 chance which one is going to be selected for the offspring. So we have 1/2. So for this, for this parent here, right? What's the probability that you're going to have the little y from the dad, right? And the little y from the dad. So that's why we set up this equation, right? The probability that you're going to get the little y from the dad is 1/2, and that's why you multiply. The probability that you're going to get the little r from the dad is 1/2. And so in total, the probability of getting the little y, little r gamete from the dad is 1/4. Okay? Now, that's just for the dad, but what about the mom? Okay, so if the mom is the same exact genotype as the dad, so big R, sorry, I erased it, little r, you can do the same steps, right? And so you're going to find that 1/4, the same chances of getting this mom is going to make a little y, little r gamete. Remember, we're looking at two different genes. So now we can finish this last piece. What's the probability that you're going to get this little y, little r from the mom and this little y, little r from the dad? Well, you already know that the probability of getting this from the dad is 1/4. So that's the dad. And then the probability of getting a little y, little r from the mom is also 1/4. So mom, dad, and mom both donating little y, little r. That's why I use the multiplication, because it's and. They're both happening at the same time. You multiply those together, you get 1/16. So when you're doing dihybrid crosses, we're going to practice and we're going to kind of come up with like routines and strategies for doing this quickly. Because I know that when you're watching this, you're like, wow, how would I do this on my own? We'll do that as a class. The second rule of probability is the rule of addition. And it's the chances that an event can occur 2 or more different ways. So let's think about an event that we can think about. And let's think about rolling a die. So let's say actually we take 2 dice and we roll them. And our goal is to get a 7. We want to, when you roll 2 die at the same time, we want to get a 7. So you could do a 6 and a 1 or a 3 and a 4, right? There's many possibilities to get a 7. But each time you're rolling, it's different. It's a different pair. So you could do, remember, 6 and 1 or 3 and 4, right? We're not saying and, we're saying or. There's different possibilities. In the same way, what's the probability if you have a dad that's heterozygous and a mom that's heterozygous that you're going to get a child that is heterozygous as well? Well, there's different ways to do it. There's different possibilities for which alleles could have been passed down to this child from this parent and which one could have passed down for this one, right? This offspring could have gotten the capital P from the mom or the dad, right? When you hear that or, I want you to think addition. So anytime you hear or, you add, okay, in these probability rules. So let's look at this problem. Probability of rolling heterozygous or having a heterozygous parent, make heterozygous offspring. So first, what you're going to do is you're going to first look at the sperm. So our goal is to get heterozygous offspring. So it's possible that the sperm could have donated for this heterozygous offspring the capital P. And what's the chances that it would have done that? It's 1/2. And that's why I use multiplication. The egg donating the lowercase p, what are the chances that dad donates capital and mom donates recessive? You multiply 1/4, right? Dad and mom. But now we have another option. What's the probability that dad will donate little p, 1/2, and mom donates capital P? The probability is 1/4. So these events are connected, right? What's the probability that you have these two events, lowercase p and capital P? It's 1/4. Okay, but now when we put it all together, you have these two options, right? What's the probability that you have this or this, right? And that is 1/4 plus 1/4. So whenever you're looking at the different ways that an event can occur, you're talking about this or this could have been the reason why we get heterozygous offspring. So we're going to add those. Okay, and we're going to come back to this later. Last thing I want to show you is eventually, we're not going to do this now, we're going to be doing chi-squared analysis on genetics problems. And why are we doing that? Well, we're trying to test if our data supports our hypothesis. So Gregor Mendel, he did experiments in the field and he actually counted up, right? If you have a purebred parent and a purebred parent and they reproduce, they cross, what percentage of the offspring look like their parents? And he collected data, but he actually used probability to check and see, is the results from like actual research, from actually collecting plants, is it similar or different from our expected, or basically what you could do with the Punnett square, predicted ratios of offspring. So remember, we're comparing our expected versus our observed data. Punnett squares make predictions based on statistics, but in reality, we might see something different. So whenever we're doing Punnett squares, we can think about your genotypic and phenotypic ratio. And so usually our null hypothesis, or H naught, is this. We want to see, our null hypothesis is that the data that we collect, right, the data that we actually collect from the experiment, in the predicted ratio, so if we predict the ratio to be A to B, is our actual experimental data going to have statistically significant difference between the observed, what we actually see out in the field, versus what you expect from Punnett squares. And the null hypothesis is that there is no statistically significant difference between expected and observed values. If there is any variance, it's just due to random chance and not some actual different effect that's being had on the population. Okay, and we'll talk more about this when we do practice problems. The alternative hypothesis is that the data does not occur in the predicted ratio. So there is a statistically significant difference between observed and expected values. And we're going to come back to this, but we're going to take a look and see maybe the reason why our observed values in the field are different than what we expect to happen is because there's possibly a different inheritance pattern. We're going to come back to this because there's different ways of inheriting alleles. The way that Mendel, or basically the research that Mendel observed, is called Mendelian genetics, but there's a whole nother type of inheritance patterns called codominance and incomplete dominance, and there's more like multiple alleles. There's more of them as well that actually has different ratios for different traits and different alleles. So we'll come back to that, but just kind of an introduction into some more statistics that we're going to be doing. Okay, last thing I want to show you, pedigrees. So we can use pedigrees, and they are actually going to show family trees, and you can actually map out what are the alleles that are found in your generation, your parents' generation, the generation before that, and you can look for patterns in inheritance and just how to read a pedigree. So here's a pedigree here, and anytime you have a square, that's going to be a male, and every time you have a female or a circle, that's going to be a female. So memorize that. That's going to be important. And when they're connected here, that means they reproduced. Okay. And then this line down here, these are the offspring of the the next generation. So here's the grandparents. They reproduce and they make the second generation. And then you have the third generation, right? Now, when you look at a pedigree, if you have a piece here that is blank, it's not filled in. Okay, that means it does not have the trait. Does not have the trait. So let's say you're looking at, for example, the trait of widow's peak. So widow's peak is here, and no widow's peak is here. Okay, so this person has the trait. Okay, so let's look at this grandparent. Notice how the grandparent is shaded in. Does the grandfather have the trait or not have the trait? Ooh, the grandfather does have the trait. And we actually see here that he does. He has the dominant and the recessive allele. Does the mom or the grandma have the trait? No, her box is not, or her circle is not shaded, right? So she does not have the trait. So little w, little w. So this shows you that the widow's peak allele is recessive, or is dominant, is dominant. So because this person does not have the trait, right, little w, they're normal. But let's see the offspring. So when you do Punnett squares and you see the offspring, these are the actual offspring that they got here is a daughter, and the daughter does have the trait, right? Because you can see capital W and little w. This one does not, this one does not. So that shows you that you actually have to have the dominant allele to get that trait. And so widow's peak is going to be dominant to no widow's peak. You can look at the same thing over here. Here's another trait, attached earlobe. So let's say you have two grandparents and they don't have the trait. Okay, but just because they don't have the trait does not necessarily mean they don't have the allele for that trait. So look at these grandparents here. Here's grandfather and grandmother. Now, if you didn't know the genotypes of like these were left away, you wouldn't really know what the genotype is. All you would be able to see is that, oh, grandpa doesn't have the trait and grandpa doesn't have the trait, right? And you can do this in your family. You can look at someone and say, oh, that's your phenotype. You don't have the trait. And then if you look at their offspring, you could actually say, ooh, there's some children of these parents that actually do have the trait. And so little f, little f, little f, little f. So that shows you, ooh, okay. So when we're looking at the trait of attached earlobe versus free earlobe, okay, the attached earlobe would be having the trait. Then in this case, this individual and this individual has a trait, but they got those alleles from their parents. And we call those parents carriers, right? Even though in this generation we didn't see the attached earlobe trait, in the offspring we do because these grandparents were carrying that allele. It was masked, though, because it's a recessive allele. And in order to have the trait, you have to have two recessive alleles. Notice that sometimes in pedigrees, in order to have the trait, you have to be homozygous recessive in this case. But in this case, in order to have the trait, you have to have one dominant allele. So you can be heterozygous or homozygous dominant. Okay, so those are kind of two different ways to represent traits on a pedigree. Now, what do pedigrees help us do? They help us understand the past and predict the future. So you can actually look at your grandparents' traits and then predict based on their alleles, based on what they look like. If you notice in the offspring that, right, even though grandma and grandpa didn't have attached earlobes, the offspring did. So that tells you, ooh, maybe they were carrying an allele. And then you can actually start predicting, okay, if this organism or this person mates with another, or this person mates with another person, you can kind of think about, okay, what is your offspring going to look like? Okay, now there are thousands of genetic disorders, and these are inherited as simple recessive traits. So you actually need to have two recessive alleles to have the trait. And so here's some examples. We're going to come back to these as well, but you have to have two recessive alleles to have it. So that's why they're more rare often, because in order to get the two recessive alleles, you have to have both parents that are carriers of the allele. And they both have to give you the recessive allele, right? If you have one dominant and one recessive, then you will not get these genetic disorders. So these require two recessive alleles from each parent, right? And so that means both parents must either have that genetic disorder or be a carrier. Okay, so these diseases are recessive. Why? It's because when we think about the chromosomes, we think about the DNA that codes for proteins that make up alleles. In these cases, they're coding for malfunctioning proteins or no protein at all. So if you were to look at the homologous pair for one of these diseases, let's say a one chromosome and one chromosome, you have the dominant allele, capital A, and a lowercase allele, little a. Well, this chromosome has the instructions for building a protein. That's working. So check, it's working. That's a check. That's a terrible check. Yes, it's working. However, if you have the recessive allele, then you actually cannot make working protein. No. Okay? And so in this case here, as long as you have the dominant allele, you're going to make some protein, right? But maybe not as much as you would if you had two dominant alleles. However, if you had two recessive alleles, if both of these were little a, then you would not be able to make working protein. And so that's going to give you that genetic disease. Okay. So we call these heterozygotes if you have one of the recessive alleles and one dominant. You're called a carrier because on the surface, you don't actually have the disease. You don't have the genetic condition. Why? Because you have one working chromosome. You have one chromosome that has the correct instructions for making the right protein that stops you from having the disease. But it's possible that if this is the chromosome, the recessive chromosome gets passed into your egg cell, and then the person that you mate with also passed down a recessive allele. Homozygous recessive offspring will have that genetic condition. Okay. So these traits are hidden. So again, like I said, let's say this parent, homozygous dominant, mates with a heterozygous parent. So they actually do have the non-working allele. So this is not working, but they have a backup, right? So you can't even see that this person has a genetic condition. So let's see what happens when they make babies. So they have some normal functioning babies here. And then here you have a carrier, right? Now they're carrying the allele like this parent. So what would happen now if you were to take this baby and you were to have it mate with another heterozygous, right? Then what you would see is the result here. So you would have a 25% chance that you're going to have homozygous dominant, so no recessive alleles, all working protein. A 50% chance, right, these two, of being a carrier, but there's a 25% chance, a small chance, that if both parents give you that little a, then you will actually have non-working protein, and then you will have that genetic condition. So that's how you can reveal these diseases and how they can come up in a population, even if your parents or your grandparents didn't even have that genetic condition, right? Because that allele was hiding in previous generations, and it was just being masked by the working allele from the dominant chromosome. All right, so that's it for chapter 11, and I will see you in class.